Okay, so it's pleasure to introduce Christian
Sorry,
Burgess,
what?
who's our speaker today. Um, as you can see from his title slide, Christian's an
I
assistant professor
probably
here.
haven't updated.
Can everyone else mute themselves, please? Thank you. Christian is assistant professor in here in the Michigan Neuroscience Institute. His primary appointments in physiology. Christian trained, did his PhD work at the University of Toronto and then postdoctoral training at Harvard, and has done quite a bit of really interesting work using a lot of cutting edge neurobiological techniques and combined with some mouse genetics to study a variety of different behavioral outputs, sleep, arousal. And he's going to talk to us today about some work he's been doing on striatal dopamine dynamics. So Christian, thanks very much. And please go ahead. And just you can take questions during the talk or at the end, whatever you prefer. So
sure, yeah, if people want to chime in, that's fine with me. All right, well, thanks for the invitation to speak today and the introduction. So yeah, I thought I would take sort of just one or two slides to introduce myself, because I think there are some folks in this group that I don't know very well. So the Burgess Lab is in the Michigan Neuroscience Institute and the Department of Physiology. And I've been an assistant professor here since 2018. And what the lab normally studies is kind of fundamental behaviors in the neural circuitry that underlie them, with a specific focus on feeding and sleep. And then we kind of take this a step further and ask how these arousal and motivational drives can affect more cognitive processing, so learning and particularly visual processing. And to do this, we use kind of the full complement of modern systems neuroscience tools, right from mapping neuroanatomical connectivity and gain and loss of function experiments using optogenetics and DREADS, deep brain recording techniques like fiber photometry and tetrad recordings, and then in vivo 2-photon calcium imaging, which allows us to look at the activity of thousands of individual neurons in mouse cortex as the animal is engaging in a particular task, sometimes over weeks or months. But actually today I'm going to talk about a bit of a side project in the lab that is interested in looking at skilled motor learning. And the reason that we even sort of began engaging in this is really from conversations that I had with Dr. Dan Leventhal, who is a colleague in neurology. He's very interested in skilled motor learning. And the fact that I had a technician in the lab, Chris Phillips, who has since left the lab and is doing grad school now, but he joined my lab to learn some of the tools and techniques that we used, but really wasn't interested in sleep or feeding research. He was really interested in kind of motor control and movement research. And so when the opportunity presented itself to kind of spearhead this collaboration with the Leventhal lab, he really kind of took it and ran with it. I'm also stealing this slide from Dan, so I'm sure some of you guys have seen this before, but so here is Steph Curry, who's a phenomenal athlete and probably the greatest three-point shooter of all time, sinking a couple of buckets in a three-point contest. And so even someone as great as Steph Curry, when he shoots the ball, he doesn't necessarily know if it's going to be a made basket or not. But when it is a made basket, he gets this kind of neural feedback in the form of an increase in dopamine that signals that this is a hit. Meanwhile, another professional athlete, but not nearly as great a three-point shooter, someone like Nick Young, can take a shot. And although, well, at first he thinks it's a hit, he then realizes that actually he's missed. And again, we'll get this kind of signal in terms of dopamine amounts that actually signal this miss with a reduction in dopamine release. And of course, dopamine does a lot of different things in the brain, some of which we are kind of traditionally interested in, and many of which relate to this kind of skilled motor learning and execution. So one is signaling reward, one is identifying or sort of assigning salience or valence to stimuli in the environment, underlying learning. And so this is this kind of reward prediction error that you see in the dopamine schematics here. And then also has a role just in motor control more broadly, so motor action, movement, vigor, and then also in arousal, which Ada Abin-Rothschild, who's a faculty member here, has done a lot of work in. And of course, dopamine's role in movement is probably sort of best underscored by Parkinson's disease. So the loss of dopamine signaling contributes to some of the movement effects of Parkinson's disease. And in plenty of studies, people have used preclinical models and animal models to show that midbrain dopamine neurons have a role in movement. And so here I just chose one example where there's a genetic knockout of dopamine signaling, and it leads to reduced movement vigor. Although there's more recent studies, a bioarchives paper from Nick Tritch's lab, for instance, that shows that perhaps actually it's not movement vigor, and really more it's that phasic dopamine is kind of permissive for movement. So I think there's still some identification of exactly what dopamine's role in movement is that needs to be figured out. But a lot of these tests don't really look at learning, nor are they looking at particularly dexterous movements, right? So a lot of the kind of movements we do, like shooting a three-pointer, involves a lot of coordination between different joints. And so that's one thing that we sort of would like to investigate in addition to the motor learning aspect. And so one way that we can do this, and this is data from Alex Bova, who is Dan Leventhal's former graduate student, is to look at skilled reaching behavior in rodents. And so in this case, this is a rat who has learned to reach for a single pellet that is elevated on this pedestal here. And you can see that one of the really interesting things that Dan Lab has developed is this ability to track the kinematics of the reach in this kind of semi-automated fashion. And Dan's lab has also shown that if you stimulate dopamine neurons as the animal is performing a reach that they've already learned, you can disrupt this reaching behavior. And so this is true of exciting midbrain dopamine neurons, in this case, specifically in the substantia nigra. And it's also true of inhibiting dopamine neurons in substantia nigra. So the next thing we wanted to figure out is when and where is dopamine released in order to facilitate skilled motor learning, and also at the level of individual trials, execution of these skilled reaches. So some of the neural circuitry involved, the sort of possible candidate regions include obviously motor cortex, certain areas of thalamus that are important for motor control, and then this kind of connectivity between these midbrain dopamine neurons, both in the ventral tegmental area and substantia nigra, and the striatum, which is composed of a couple of different areas I'll talk about in a minute. And so really that's where we kind of honed in, is this connectivity between midbrain dopamine neurons and the striatum. And so here you have kind of a cartoon of that connectivity. So as you move from these medial ventral tegmental area dopamine neurons that tend to project to the more ventral parts of the striatum in the nucleus accumbens, as you move more laterally, you get into substantia nigra, which provides dopaminergic input to dorsal medial and dorsolateral striatum. And these kind of subregions of the striatum are important because they underlie different behaviors, and dopamine probably has very different effects within these areas. So this is a bit of a simplified schematic, obviously, but the general idea is that more ventral areas of the striatum in the nucleus accumbens are more important for things like assigning value and driving motivation and this kind of like Pavlovian learning. And then as you move more into dorsal medial striatum, it's more kind of goal-directed movements. And then dorsolateral striatum is more sort of habitual movements. And these areas have different connectivity with other brain areas outside of the striatum. So we aim to identify the endogenous release dynamics of dopamine in different parts of the striatum across both learning and execution of skilled reaching behavior, and to identify the functional role of dopamine release in these specific subregions of the striatum. So to do that, we started by using a fluorescence-based dopamine sensor, in this case, one called D-Light 1.1. Basically what this is a GFP molecule tagged onto a dopamine receptor. And this can be expressed in neurons within the striatum such that when dopamine is bound to this receptor, it allows for the fluorescence of green light, or the emission of green light, which we can then collect and basically get an online readout of dopaminergic tone in a given area. And so we combine this with fiber photometry, where we put an optic fiber deep into the brain. And this way we can record dopamine release in specific areas with very fine temporal and spatial resolution. And so here's just a simple Pavlovian feeding task to kind of give you a sense of what this looks like. So here we showed a visual stimulus for 3 seconds and then gave a reward, an ensure reward. This was to food restricted mice. And so in green here is the dopamine signal, or the delight signal. And what you can see is that in naive mice that have not yet learned the association, there's very little dopamine response to the visual stimulus, and you only get a dopamine response when the animal begins to lick for the reward. Meanwhile, in well-trained animals, you get this robust increase in dopamine at the onset of the visual stimulus, despite the fact that the reward is not given until the end. And so this is that reward prediction error, where you get a shift in the dopamine signaling from the reward to the thing that predicts the reward. And is also just a good illustration of how well some of these dopamine sensors work. And so this allows us to track dopaminergic tone in the same animals over long periods of time, which facilitates investigating more complex forms of learning and more kind of slowly changing behaviors than what you see here with a simple Pavlovian paradigm. And so we're now using this approach to look at, a number of different roles for dopamine, including looking at dopamine release across sleep-wake cycles to try and investigate some sleep disorders that we're interested in. But for today's talk, we're specifically interested in dopamine release within the striatum. So we injected D-light into the striatum, put a fiber optic above the area of the injection, which allowed us to deliver excitation light and collect the emitted light, which is our sort of index of dopaminergic tone. We put fibers throughout the striatum. We'll divvy that up into different areas in a bit. And again, I just want to highlight that this is work done in collaboration with Dan Leventhal, and his work almost entirely done by Chris Phillips, a technician in the lab who's now a graduate student at UT Dallas, and Courtney Myers, who is another a master's student and technician in the lab. So we then took these mice, you can see the fiber plugged in here, and we taught them how to perform a head-fixed skilled reaching task. So here you can see the animal reach out, grab the pellet, and then consume it. And so what this paradigm looks like is a mouse head-fixed in front of a wheel that delivers pellets. And we also have 2 high-speed cameras that are acquiring at 240 frames per second so that we can score the animal's behaviors. And so on each trial, an LED illuminates to cue that the trial is about to begin for the mouse. Then there's a stepper motor that's triggered to put a pellet in front of the animal. And at this time, we start doing our video recording, and then the pellet is available about 2 seconds after the LED starts. So this is an individual trial, and mice will do 20 trials per run and up to 4 runs a day with variable learning rates. And so here you can see some of the learning. Each individual dotted line is 1 mouse, and then the black is the average. So mice will increase their attempt rate over time. They will increase their success rate over time, with the average reaching about 60% success. And they'll also decrease their latency to the reach over time. So mice can get quite good at this task, although it can be a bit of a challenge to train them. And so we hypothesized that dopamine release in the striatum would be elevated during execution of skilled movements, that dopaminergic dynamics in these different striatal subregions would be different, and that dopamine dynamics across learning would be different. So here, just to kind of orient you, at the top here, you have the picture of the mouse at the point in the task that we were looking at. And so in this case, this is the response to the trial onset cues, so the LED and the stepper motor. And at the bottom, we have the D-light signal. So in green is the dopaminergic tone, and in black is a GFP control. So we wouldn't expect there to be any dynamics here other than, for instance, movement artifacts potentially. And so here you can see that there's a robust increase in dopamine to the LED as well as to the stepper motor, which are both predictors of whether a pellet is going to be available. When we now look at the start of reach, you see this very robust increase in dopaminergic tone, basically starting at reach onset. This continues through pellet contact, so when the animal actually grabs the pellet, and seems to peak right around the time that the animal starts consumption of the pellet. So just looking at group data here, you can see that there's a significant increase to these trial onset cues and a significant increase at the reach onset that is not present in the GFP controls. So importantly, it's not just a sort of movement artifact, but really is a measure of dopaminergic tone. We did this both in males and females, and showed that there's really no differences in dopaminergic tone during reaching, and also that they're both learn at similar rates. And then in a subset of mice, we also did this bilaterally. So looking at dopamine release in the contralateral striatum and the ipsilateral striatum to the mouse's preferred reaching paw. And so what you can see here is actually there's no difference really between the contralateral and ipsilateral dopamine signaling. And that's true in the sort of group data here as well. And this actually kind of surprised me at first. I had expected, perhaps naively, that you would have greater dopamine release in the striatum contralateral to the reaching paw. But it's possible that, either that the dopamine is released bilaterally and it's not doing much because there are some dopamine neurons that project to both striatum. But it's also possible that, especially for a mouse where it's reaching out with one paw, but is kind of bracing itself with its other paw, that's actually a part of the reach, is the sort of bracing of itself. And so just in a subset of mice, this was a difficult experiment to do because mice don't train very well with their less preferred reaching paw. But Chris trained mice to reach with their preferred reaching paw. They got quite good at the task. And then he basically blocked that reaching paw and forced them to use their less preferred one. And again, they can perform the task quite well. And when we look at contralateral versus ipsilateral dopaminergic tone, you see that there's really no difference even after forcing mice to use their less preferred paw. So I had said in the introduction, there is this distinction between different parts of the striatum. And up to this point, we've kind of been lumping all of those together. But certainly with a task like this that involves learning, but also motor action and goal-directed behaviors and other things, that we thought it would be interesting to look specifically at these different parts of the striatum. And people have shown before that there are pretty diverse dopaminergic dynamics across the striatum. So here's a study from Markow looking at photometry, but instead of looking at dopaminergic tone here, he's actually measuring the activity of dopaminergic terminals in the striatum. And what they show is that in the dorsal striatum, when you record, there is a strong response of these dopamine terminals to acceleration, but not so much to reward. And then as you move to the more ventral aspects of the striatum, you now have much less response to acceleration, but this very robust response to reward. And so in a small subset of mice, we basically replicated this, where in the dorsal striatum, which are the recordings on top, we see very little response when the animal licks for a reward, but an increase when the animal is running on a running wheel. And then in the ventral striatum, we see this very robust increase when the animal is licking for a reward, and much less of a change when the animal is running on a running wheel. So this just underscored the importance to us of separating out our recordings that were in different areas, different parts of the striatum. And so we did this, and we investigated dopamine release in the dorsolateral striatum, which is this kind of dark blue color, the dorsal medial striatum, which is this purple color, and then the ventral striatum, which is this orange color. And so we'll look at each of those for the rest of the talk separately. So here we're just looking at dorsolateral striatum. And again, this is the time course of dopaminergic tone. And on the bottom, we have hit trials. So this is where the animal was successful in consuming the pellet. And at the top are miss trials. So these are ones where the animal attempted a reach and even potentially sort of made contact with the pellet, but wasn't able to grasp the pellet and bring it back to its mouth and actually consume it. So in a way, this hit is similar to the Steph Curry GIF at the beginning of the talk, and the miss is more of the Nick Young GIF. So here you can see in dorsolateral striatum there is a small increase in response to the cues that predict trial onset, and a very large amplitude and fast dynamics dopaminergic release at the onset of reach. And this persists through pellet contact and consumption. And so if we look at the group data, you see a significant increase to cues that predict the trial to the onset of the reach. And then in pink here is a comparison between hit and miss trials during this pink shaded box. And so this is that period where the animal has sort of up to this point made the same or very similar motor action. But now in the miss trials has realized that they're not going to get a pellet, whereas in the hit trials, they realized that they are going to get the pellet. And so this is a readout of this reward prediction error. And you see it quite clearly even in dorsolateral striatum. Now moving on to dorsomedial striatum, you see this much more robust increase in response to trial onset cues. Still a good increase in response to the start of the reach, albeit lower amplitude and more kind of spread out. And if you look at the group data, again, you see that robust increase to the cues, robust increase to the reach, and again, this sort of marker of reward prediction error, where when the animal doesn't get the pellet, you get this dip in dopamine. And now into ventral striatum, where again, this sort of trend continues, where you have now even stronger responses to the trial onset cues, still responses to the reach, although they're a little muted relative to other areas. And again, and of course, we would definitely expect this within the ventral striatum, but these reward prediction error-like signals. So if we just sort of overlay those three traces, so again, in blue is dorsolateral striatum, purple is dorsomedial, and in orange is the ventral striatum. You can see that there are various differences across time between these three groups, but really the most striking thing is this very high amplitude, fast dynamic dopamine release within the dorsolateral striatum. So the next thing we wanted to do was look across learning, right? So even in this kind of simple Pavlovian learning that I showed at the beginning of the talk, it's very clear that dopamine dynamics can change across learning. And because of the nature of D-light signaling and the fact that it can take mice a little while to learn this task, we could actually track the dopamine signals across time as the animal increased their success rate. And so here are examples from individual animals. So just to orient you, if we just focus on this upper left box. So this is a heat map where every line is an individual hit trial from one animal. And the darker the color, the more dopamine activity that was present at that time. And the dotted line signals trial onset. In the right, the dotted line signals the start of the reach. And so what you can see is that in the dorsolateral striatum in blue, you can see kind of the emergence of these slight dopamine responses to the trial onset cues. But what really jumps out is how regular and robust this increase in dopamine at the start of the reach is. And it sort of persists even once the animal is an expert towards the end of the reaches here. But then when we look at dorsomedial striatum and perhaps even more strongly in ventral striatum, you see that it starts out with this very robust dopamine release at reach onset, but that quickly transitions over into these trial onset cues. And actually the reach response is really diminished. And especially in the ventral striatum, as you get to the end, the reach response is almost gone.
So Christian, do you have any explanation for why that two peaks in the ventral striatum seems to be so prominent? One associated with the cue per se and the other with the motor, and it looks like it gets attenuated in dorsal medial, but it's really strong in the ventral.
Yeah, no, I mean, I don't totally know it. And frankly, I sort of wish that, when we designed this task, we weren't sort of purposely having these two separate things that cued the trial. It just turns out that since we need to have a motor and the motor makes some noise and also the mouse can see the thing start to turn, that it has this kind of second association. There are previous studies that sort of look at adding, cues that predict cues and sort of dopamine's role in that. And at some point, we should probably go and look at that and see if we can, sort of figure out what's underlying some of this shift.
Yeah. Well, I think the folklore is that Wolfram Schultz discovered RPEs supposedly by the fact that there was an unanticipated cue in the gizmo that was delivering pellets to his monkeys, and it was the sound of the monkey when the what they thought was going to be a reward signal, it migrated to when the motor went off in the pellet delivery apparatus. So.
Right. Yeah, no, and I mean, even in my other work where we do visual association stuff, I've seen that where the sound of the solenoid that's delivering the liquid reward will be enough to link to it, and they'll kind of disregard the visual stimulation entirely. So, but yeah, that's definitely something that we want to kind of think more about here, and probably in future experiments try and find a way where we can combine those cues together such that we're not having this kind of biphasic response. Okay, so now, again, we're looking across learning now just in dorsolateral striatum. So just to orient you, the light color here, the light blue, is early learning trials, and the dark blue is late learning trials. And importantly here, we're only looking at hit trials. So even though this mouse is early in learning, every trial that is incorporated into this trace and into the group data is a trial where the animal got the pellet. And so again, here you see the emergence of these kind of small responses to the cues, albeit significant. But then what's interesting is that when you look at the start of the reach and the consumption triggered time courses, you see that they're basically perfectly overlapping. So there's not really any change in the dopamine release at reach onset in dorsolateral striatum, even as the animal gets much, much better at the task. And so we quantified a few other things like the peak dopamine release, the half peak width, as well as the peak time, all triggered to the start of the reach. And none of these things are significantly different across learning. When we look at dorsal medial striatum, again, now you can see this very clear emergence of the cues that predict the trial onset. And now when we look at reach, we do see a reduction. So again, the light color here is early learning, the dark color is late learning. We see a reduction in the dopamine signaling at reach onset, and it seems to sort of be specifically at this time of consumption. And again, that's reflected in some of these other measures like the peak dopamine amount and the time of the peak shifting a little bit early. And then in ventral striatum, again, this even more striking sort of emergence of trial onset cue responses. And here you really see this suppression of the reach response pretty strongly. Again, sort of totally in line with reward prediction error. And again, that's reflected in some of this average data. And so here I've just kind of put all of the data on one slide so that you can kind of look at it all together, although it's maybe a little overwhelming. But in general, you kind of see this pattern of, very little change, especially during the reach in the dorsolateral striatum, more of a change in dorsal medial, and then a more striking change even in ventral. And again, I think that there's probably more work we can do here to get at some of the sort of richness of the way that the dopamine pattern changes across learning. And actually, I'll get to sort of one idea that we're doing in a minute to get at that. But so just to sort of conclude that part of the work. So dopamine release in the striatum is increased at reach onset, and it generally peaks around the time of pellet consumption. The dopamine levels are very similar in ipsilateral and contralateral striatum. So sort of regardless of how the animal or what paw the animal is reaching with, dopamine release seems to be pretty similar bilaterally. The dopamine dynamics show this robust reward prediction error in miss versus hit trials, and it's pretty much regardless of what area of the striatum you're in. And that these dopamine dynamics change across learning with much greater changes happening in more ventral aspects of the striatum than dorsal. So I'd mentioned this at the beginning that Dan's lab has developed these really nice, robust tools to track reach kinematics, which I think is especially important when we're looking at something like a dexterous movement that requires a lot of orchestration across different joints. And so, you know, they did this initial study that I'd mentioned previously, but Alex and Dan have done a follow-up study where they've looked across learning. And for instance, one of the things I think is really interesting from this is just you can basically show that these kinematics get a lot more regular. The reach trajectory gets a lot more regular as the animal gets better and better at the task, which makes a lot of sense. But I think this can also be an interesting tool for us to look at what I just mentioned of the kind of richness of how the dopamine signaling changes over time as the animal learns. And so if we can sort of put together the dopamine dynamics along with the individual reach kinematics, then we can start to ask questions about whether one, you know, leads or lags the other. And so we're doing this now. Here's just an example of some preliminary deep lab cut based tracking of reach kinematics in the mouse. So you can see that the semi-automated sort of scoring of this has identified the ears, the eyes, the nose, and then the individual digits on both hands of the mouse, as well as the pellet. And so by using this, we can track in 2 dimensions the kind of trajectory of the reach. But because we have 2 cameras, and this is something that Dan is helping us out with now, we can actually recreate this trajectory in 3 dimensions. And so we're now doing this, and we've already got all of the dopamine data analyzed. So we're going to kind of correlate those 2 things, and as I said previously, basically see if the changes in dopamine dynamics kind of underlie these improvements in reach kinematics, or at least more and more regularity in the reach kinematics. I'd also mentioned before that Dan has done this gain and loss of function at the level of substantia nigra dopamine neurons, and shown that you can disrupt an already learned reach. And so we'd like to kind of extend this now and think about not just an already learned reach, but are there different times during training that we can stimulate to disrupt learning rather than just execution of the reach. And then also looking at, again, these specific areas of the striatum. So substantia nigra obviously predominantly projects to dorsal striatum and not to ventral striatum or motor cortex. So we'd like to sort of go in with more granularity and do terminal stimulation or inhibition to be able to look at the role for different parts of the striatum. And so we've done some very kind of preliminary work in that regard, using a retrograde Cree approach to get expression of red-shifted opsins into midbrain dopamine neurons, and then also recorded D-light signaling as we did that. So here's an optic fiber down into nucleus accumbens core, and in green you can see the D-light expression, and in red are terminals from midbrain neurons. And now those of you that kind of work with viruses may have already noticed that this is not going to give us dopamine specific labeling. It's just giving us midbrain neurons that project to the striatum labeling. And so there's probably a mixed population that we're stimulating here. And so we're actually breeding up DAT-Cree mice to get more specific versions of this experiment. But we can show that we can stimulate terminals within the striatum and simultaneously record dopaminergic tone and show that we get this evoked dopamine release and that it scales with the intensity of the optogenetic stimulation. And so we've done this in a few mice, but it's been entirely ventral striatum and entirely post-learning. And actually what we see is that it doesn't disrupt the reaching at all. So the effect that Dan showed at the level of the substantia nigra at least doesn't sort of persist in ventral striatum. I suspect if we were to stimulate terminals in dorsal striatum, we probably would see much stronger effects and disrupt this reaching. And then, of course, we'd like to do that in learning as well. And so these are things that are ongoing right now. But in future, we have plans to kind of use these same tools to investigate the circuitry that underlies skilled motor learning and execution in some mouse models of movement disorders, so Parkinson's disease and dystonia. We'd also like to extend this approach to looking at cholinergic dynamics in the striatum. This is something, again, we're currently kind of piloting now, but there are these cholinergic intraneurons within the striatum that play an important role in coordinating dopamine release. And then in the more kind of medium to long term, something I'd be very interested in doing is combining this sort of behavior and training and also automated tracking with some of this large scale recording techniques that we use to be able to look at whether some of these same dopamine dynamics that we're looking at in striatum are also modifying the kind of like reach ensemble within motor cortex. And so these are kind of more longer term future goals. And with that, I just like to acknowledge my lab, especially Chris Phillips, again, who is unfortunately now in graduate school at UT Dallas instead of Michigan. Courtney Myers, who is just applied to Michigan for graduate school this year. And Elizabeth Pappas, who's an undergraduate student in the lab that's been working on some of this stuff. Also, obviously, acknowledge Dan Leventhal and Alex Bova and the rest of Dan's lab, who's been great in helping us get all this stuff up and running. And then funding, but especially the Brain Research Foundation, which funded the work that Chris did on this. So with that, I will take any questions.
Okay. Okay, so we're open for questions. So do you think that cholinergic interneurons are going to modulate this differentially in different regions?
That's an interesting question. I don't know, to be honest.
Yeah.
We're kind of excited to try. So we've got these cholinergic sensors that we haven't used much in-house, but I have some colleagues that have used them and they seem to work great. And the idea is to sort of combine dual sensors along with optogenetic activation of either dopamine or acetylcholine and being able to get to the bottom of that sort of relationship.
Okay. All right, very good. Any other questions? Okay. Any other questions for Christian today? Okay. Well, in that case, thanks very much for a really nice talk. And that was quite interesting. We'll have to have you back, when this is close to completion for an update.
Yeah, no, it would be nice to have the DSC data and the optogenetics data all finished.
Yeah.
Hopefully soon most of that will be done.
Okay, very good. All right, thanks everyone for attending today.
Thanks, guys.
Us now. So.
I don't know why this.
Okay.